Welcome back, everyone. I am absolutely thrilled to have you here for this explainer. Because today, we are going on a guided tour of the ultimate, most sophisticated chemical processing plant in the known universe. Yep, the human body. We're going to break down the incredibly complex, beautifully orchestrated systems that keep us safe from the myriad of foreign substances we encounter every single day. So get comfortable and let's decode the biology of you. It's a simple question, right? What actually happens to a pill after you swallow it? Or really any foreign chemical that enters your system? Our bodies are constantly tackling this major problem, how to eliminate foreign chemicals, what biologists call xenobiotics. Now, the human body is mostly water, so getting rid of water-soluble chemicals, that's pretty straightforward. But the real challenge, the ultimate puzzle our bodies have to solve, is what to do with fat-soluble or lipophilic chemicals. If we can't process them, they stick around. And honestly, that is exactly where the danger lies. So here's the blueprint for our factory tour today. 1. The body's chemical factory. 2. Phase 1, the modifiers. 3. Cytochrome P450, our superstar. 4. The genetic variable. 5. Esterases and prodrugs. And finally, 6. Phase 2, final packaging. Let's break this whole thing down.
Okay, let's dive into section 1, the body's chemical factory.
When we hear the word metabolism, we usually just think about, burning calories or digesting our food. But in the world of pharmacology and toxicology, metabolism has a very specific, absolutely critical definition. It's this highly energy-intensive biotransformation process. Its entire purpose is to reduce those foreign invaders, those xenobiotics, by forcefully increasing their water solubility. It's all about taking lipophilic, fat-loving invaders and basically forcing them into a water-soluble format so they can be safely flushed away by the kidneys. The ADME blueprint perfectly illustrates exactly where metabolism fits into the whole disposition picture. ADME stands for absorption, distribution, metabolism, and excretion. Once a drug is absorbed into your systemic circulation, it distributes out into your tissues. But here's the kicker. If metabolism doesn't step in, the drug just keeps cycling back and forth into your tissues indefinitely, which can easily reach toxic concentrations. Metabolism is the vital intervention that intercepts the drug, alters it into a metabolite, and redirects it straight toward the exit door for excretion in urine or feces. Moving on to section 2, phase 1, the modifiers. As we kind of touched on a second ago, the body definitely plays favorites based on chemical structure. If a chemical enters your body and it's already water-soluble, well, it gets a fast pass. It enjoys rapid elimination. But those fat-soluble chemicals, they face something called sequestration. Without metabolism, they will literally hide out and accumulate inside your fat and tissue cells. Over time, that sequestration leads to severe toxicity. So phase 1's entire job is to prevent that sequestration from happening by physically altering those stubborn fat-soluble molecules. So how do they actually pull this off? Well, phase 1 reactions, which are also known as functionalization, are where the magic really starts. These phase 1 enzymes act as the ultimate molecular modifiers. They force chemical reactions, things like oxidation, reduction, or hydrolysis, to introduce what we call polar functional groups. Think of groups like an OH group or a COOH group. You can just imagine these groups as tiny molecular handles. By attaching these polar handles to a smooth, fat-soluble chemical, phase 1 preps the molecule, basically giving the next team on the assembly line something to grab onto. Which brings us to section 3, cytochrome P450, the superstar. You're going to see the acronym CYP a lot in pharmacology. So let's just decode the jargon real quick. Cytochrome P450s are this massive multifamily group of enzymes, and they have a highly structured naming convention. The first number, like the 1 in CYP1, tells you the broad family. The following letter, like an A, denotes the subfamily. And then that final number pinpoints the exact specific enzyme, like CYP1A2. Now, while there are a ton of families out there, families 1, 2, and 3 are absolutely the heavyweights when it comes to human drug metabolism. Now, what's really wild here is the sheer scale of the work these specific enzymes do. If you look at the breakdown of all phase 1 enzymes in the body, there is one massive share that belongs to just CYP3A4 and 3A5. That single subfamily handles the metabolism for a staggering percentage of all the prescription drugs on the market today. I mean, it perfectly proves just how much heavy lifting the cytochrome P450 family does to keep our internal factory running smoothly and safely. Let's actually look at these factory workers in action. Take diazepam, for example, a really common, highly lipophilic anti-anxiety medication. When diazepam hits the liver, those CYP450 enzymes get right to work. They completely transform the molecule into a metabolite called oxazepam. The CYP enzyme physically modifies the original structure, attaching a new polar handle right onto the ring. The drug has been functionalized, it's fundamentally changed, it's prepped, and it is so much closer to being easily excreted. Mechanically, how is this even happening? It's honestly a microscopic marvel. First, these CYP enzymes are physically embedded right in the lipid biolayer of the endoplasmic reticulum, which is basically the inner walls of your cells. Once a lipophilic drug substrate binds to the enzyme, the CYP complex utilizes this incredibly precise electron transfer, handed over by a helper molecule called NADPH. Using that electrical energy, the enzyme forces an oxygen atom directly into the drug molecule. It is a highly coordinated, energy-intensive process that is happening literally millions of times a second in your liver. Crazy, right? All right, section 4, the genetic variable. This brings us to a rapidly growing field called pharmacogenetics. And I want to slow the pace down here just a bit to really emphasize this because it is absolutely crucial to individual health. Pharmacogenetics is the assessment of how your specific, completely unique genetic makeup dictates your drug metabolism. We are not all issued the exact same factory equipment at birth. The genes you inherit determine exactly which versions of these CYP enzymes you actually have, and consequently, how well they work. The real-world stakes of this genetic variation are just massive. Because of genetic differences in CYP enzymes, giving the exact same dose of the exact same drug to two different people can yield wildly different results. For one person, it acts as a perfect healing drug. For someone else, say someone with an ultra-rapid metabolism, the drug might be cleared out before it even has a chance to work, making it completely ineffective. And in someone with a poor metabolism, the drug could just build up and create toxic metabolites. And actually, allele frequencies for these enzymes can shift dramatically across different populations. For instance, we see really stark contrasts in drug handling between Ethiopian and Saudi Arabian demographic groups. Essentially, your genetics are the ultimate floor manager of your metabolic factory. Let's jump to section 5, esterases and prodrugs. Now, CYPs don't do all the work alone. Esterases are specialized enzymes that catalyze the hydrolysis of specific chemical bonds. We have carboxyl esterases stationed in the liver, the kidneys, and the small intestines, just waiting to break down incoming molecules. And then we have cholinesterases, which are super involved in metabolizing neurotransmitters. For example, acetylcholinesterase operates in your red blood cells and central nervous system, while butyrylcholinesterase patrols your blood plasma. They have these incredibly specialized, highly targeted roles in managing both our internal chemistry and outside drugs.
In understanding these specific esterases has actually led to one of the most brilliant innovations in pharmacology, the prodrug. A prodrug is essentially a chemical Trojan horse. It's a medicine you swallow that is entirely inactive. It does literally nothing on its own. But it is chemically designed so that once it meets a specific esterase enzyme inside your body, the enzyme cleaves off a piece of the molecule, instantly activating the drug right exactly where it needs to be. We are literally using the body's own factory workers to unpackage and activate the medicine. How cool is that?
And finally, section 6, phase 2, final packaging. Phase 2 is all about conjugation. We take that modified phase 1 metabolite with its newly attached polar handle, and we hook a massive chemical trailer onto it. Using electrophilic cofactors, phase 2 enzymes attach large, bulky functional groups, things like sugars, acetates, or sulfates. By strapping this massive, highly polar tag onto the molecule, we achieve two crucial things: a very high molecular weight and complete, total water solubility. The chemical is now fully neutralized, all packaged up, and entirely ready to be safely excreted from the body. The factory has done its job. So, we've seen how beautifully complex this whole system is, from the CYP superstars to the heavy-lifting conjugation enzymes. But think back to that genetic variable we talked about earlier. Our bodies possess this incredible, universal chemical disposal system, yet each of ours is tuned just slightly differently. As we map more and more of our genetic code, how might deeply understanding your unique individual chemical factory completely change the future of customized medicine? Imagine a world where your prescriptions are tailored perfectly to your specific CYP enzymes. It's an incredible frontier. Thank you so much for joining me on this tour of the human body's metabolic factory. Keep learning, keep questioning, and I will see you in the next explainer.